All. So today's topic of discussion is orotic aciduria. Now come firstly the introduction. So it is an autosomal recessive disorder. Autosomal recessive disorder, and it is also known as hereditary orotic aciduria because a variation in the UMPS gene, that is uridine monophosphate synthase gene, UMPS gene, a variation in this gene can cause orotic aciduria. It is a disorder of de novo synthesis of de novo synthesis of pyrimidine. The enzyme that is deficient or defective is uridine monophosphate synthase, that is UMPS. So it is a bifunctional enzyme and it has two activity, that is OPRTase activity and OMP decarboxylase activity, that is orotate phosphoribosyl transferase. Orotate phosphoribosyl transferase OPRTase activity and orotidine monophosphate or OMP decarboxylase activity. So the OPRTase is the enzyme of the fifth step and OMP decarboxylase is the enzyme of sixth step of de novo synthesis of pyrimidine. So this is the fifth step. Orotic acid in presence of OPRTase becomes orotidine monophosphate or orotidylic acid, OMP. The enzyme is OPRTase, and in this reaction PRPP, that is phosphoribosyl pyrophosphate, phosphoribosyl pyrophosphate, phosphoribosyl pyrophosphate is converted into inorganic phosphate or pyrophosphate. So orotic acid in presence of OPRTase becomes OMP, orotidine monophosphate, and in this reaction phosphoribosyl pyrophosphate becomes pyrophosphate, that is PPI. Now coming into the sixth step, this is the sixth step of de novo synthesis of pyrimidine. So OMP, that is orotidine monophosphate, becomes uridine monophosphate, that is UMP or uridylic acid in presence of UMP decarboxylase. So OMP becomes UMP in presence of OMP decarboxylase. So the enzyme is decarboxylase, so a molecule of CO2 is eliminated from OMP and thus we get UMP. So this is the sixth step. Now this orotic aciduria has got two types, that is type 1 and type 2. In type 1, both the enzymes, that is OPRTase as well as OMP decarboxylase, both these enzymes are deficient. So that is type 1, and in type 2 only OMP decarboxylase is deficient. So in type 1 both the enzymes OPRTase as well as OMP decarboxylase or OMP decase in short. So OPRTase as well as OMP decase both are deficient in type 1 and only OMP decarboxylase is deficient in type 2. So that is about the introductory part of orotic aciduria. Now coming into the features. So accumulation of orotic acid crystals are seen. In the fifth step OPRTase, this enzyme is deficient, right? So this step will not take place. So this orotic acid will get accumulate in our body. So that causes accumulation of orotic acid crystals. And these are the intermediate steps in the de novo synthesis of pyrimidine. So if these steps, these fifth and sixth steps are blocked, then that causes an obstruction in the synthesis of pyrimidine, and that causes that results in the impaired pyrimidine synthesis, and that causes defect in DNA and RNA synthesis. So a defect in DNA and RNA synthesis is seen in orotic aciduria due to the impaired pyrimidine synthesis. And that is, we already told, the OPRTase and OMP decase enzymes are deficient. These are the enzymes of fifth and sixth steps respectively. So these steps will not take place, and that obstructs the de novo synthesis of pyrimidine. So that is the features of orotic aciduria. Now coming into the clinical manifestations, that is the signs or symptoms that are seen in the individual with orotic aciduria. First one, it is the retarded growth, then megaloblastic anemia, orotic acid crystals in urine, and urinary tract obstruction. So the first and second, that is retarded growth and megaloblastic anemia, that is correlated with the defect in DNA and RNA synthesis. So that causes retardation in the growth. And megaloblastic anemia, we all know it is the presence of abnormal precursors of erythrocytes in the bone marrow, that is megaloblast. So it is due to the defect in DNA and RNA synthesis in these precursors of erythropoiesis. So that causes megaloblastic anemia. Now retarded growth, okay, that we discussed. Now then it is orotic acid crystals in urine. So the name of the disease itself is orotic aciduria. Aciduria. So aciduria means presence of something in the urine. So orotic acid is present in urine. So it is because the accumulation of orotic acid in body, and this symptom is very evident from the name itself. So as the orotic acid crystals are seen in urine, there is a high chance for urinary tract obstruction. So urinary tract obstruction is seen in orotic aciduria. So these are the clinical manifestations. Now coming into the diagnosis, there are mainly 4 diagnoses that we usually use. First one is urine analysis. That is to identify if orotic acid is present in the urine. That is urine analysis. Then bone marrow screening. It is to check if megaloblasts are present in bone marrow. That is, it is usually used for detecting megaloblastic anemia. But as megaloblastic anemia is a manifestation of this orotic aciduria, we can use bone marrow screening also as a diagnostic technique. The next technique is gene testing. It is to check if there is any variations in the UMPS gene, that is uridine monophosphate synthase gene, the gene which is responsible for the enzyme activity of UMPS, enzymatic activity of UMPS. So that is gene testing. Fourth one is enzyme assays, that is to check for the presence or if the OPRTase and OMP decarboxylase are deficient in the body. So that is enzyme assays. Enzyme assay for OPRTase or orotate phosphoribosyl transferase, that is OPRTase, and orotidine monophosphate decarboxylase, that is OMP decarboxylase. Okay, so these are about the diagnosis of orotic aciduria. Now coming into the treatment part, supplementation of cytidine and uridine are the main procedure of treatment. So this cytidine and uridine, it forms CTP and UTP respectively. Cytidine forms CTP and uridine forms UTP. It is by the action of kinase enzymes. The kinase enzymes like cytidine kinase, uridine kinase, CMP kinase, UMP kinase, like that many kinase enzymes act, and finally we get UTP and CTP. The kinase means we all know it transfers one phosphate moiety from ATP to the substrate. So here the substrate is cytidine or uridine. So it takes one phosphate group, transfers one phosphate group from ATP to this cytidine and uridine, forming CMP and UMP, and subsequently we get CDP, then CTP. Similarly in UMP we get UDP and UTP. Okay, so this UTP and CTP, these are the final pyrimidines that is required for DNA and RNA synthesis. So the DNA and RNA synthesis which was obstructed due to the deficiency of OPRTase, and OMP decarboxylase enzyme, is now revived with the supplementation of UTP and CTP. So this UTP and CTP enters into the DNA and RNA synthesis, and thus DNA and RNA synthesis is revived and it becomes normal. So the manifestations due to the defect in DNA and RNA synthesis, that is retarded growth, megaloblastic anemia, are all cured with this cytidine and uridine supplementation. And another action of UTP is that UTP is a allosteric inhibitor of carbamoyl phosphate synthetase 2. Carbamoyl phosphate synthetase 2. The reason why I said 2 it is because carbamoyl phosphate synthetase, that is CPS1, it is an enzyme of urea cycle, and it has different function, but it causes the formation of carbamoyl phosphate, but it is the formation of carbamoyl phosphate is from another compounds that is discussed in urea cycle. So in de novo synthesis, it is carbamoyl phosphate synthetase or CPS2. Okay, so this enzyme is the enzyme of first step of de novo synthesis. The first step of de novo synthesis is described here, that is bicarbonate in presence of CPS2 gives carbamoyl phosphate. So in this reaction, a molecule of glutamine is converted into glutamate, 2 molecules of ATP is converted into 2 molecules of ADP. So this is the first step of de novo synthesis. So if the CPS2 enzyme is allosterically inhibited by this UTP, then this step will not take place. This step will not take place. So this step is inhibited, and that causes a decrease in concentration of carbamoyl phosphate. So the carbamoyl phosphate is the product of first step, and it enters into the second step of the de novo synthesis. If carbamoyl phosphate is reduced, then the subsequent steps of de novo synthesis will not take place. Thus, the concentration of orotic acid also decreases because the subsequent steps of de novo synthesis will not take place. So orotic acid will not get accumulated in our body. So no aciduria is seen in that individual. So even though the de novo synthesis is inhibited by UTP, the DNA and RNA synthesis are revived due to UTP and CTP, that is supplemented to the individual. So the cytidine and uridine supplements is like a really curative treatment method. So that is about the treatment. Now other types of orotic acidurias that are seen. First one is Reis syndrome or secondary orotic aciduria. It is due to the deficiency of ornithine transcarbamylase or OTC enzyme. Ornithine transcarbamylase. This is the enzyme. Ornithine transcarbamylase is an enzyme of the second step of urea cycle. Second step of urea cycle. The second step is ornithine plus carbamoyl phosphate gives citrulline. So this OTC is deficient, so this step will not take place. So carbamoyl phosphate will get accumulated in our body. So the more amount of carbamoyl phosphate generates, so the accumulated carbamoyl phosphate enters into the de novo synthesis, and the orotic acids are produced excessively. But in this OTC deficiency, the enzymes, that is the OPRTase as well as the OMP decarboxylase, these are not deficient or defective. So that enzymes are highly active. Those are active in their normal state. So the orotic acid which are produced excessively are subsequently converted into the next products of de novo synthesis. So that's why the orotic acid will not get accumulated excessively in our body. So that is why it is a mild form of orotic aciduria. This Reis syndrome is a milder form of orotic aciduria. So that is secondary orotic aciduria. Now another one is drug-induced orotic aciduria. It is due to allopurinol, a drug known as allopurinol. It competes with orotic acid for the OPRTase, the active site of OPRTase. So the orotic acid will not get converted into OMP, that is orotidine monophosphate. So the conversion is reduced, and thus orotic acid will get accumulate in our body, and also the pyrimidine synthesis will get suppressed or it reduces. So it causes orotic aciduria. So that is the drug induced orotic aciduria. Now about the differential diagnosis, that is to identify what is the cause of the orotic aciduria. Now we discussed three causes, right? One is due to the deficiency of UMPS, that is uridine monophosphate synthase enzymes, that is OPRTase enzyme and OMP decarboxylase enzyme. That is the first cause. The second cause is we discussed the Reis syndrome, that is the deficiency of ornithine transcarbamylase. The second is due to the drug allopurinol. So to identify what is the cause of our orotic aciduria, we use differential diagnosis. First one is blood ammonia level and blood urea nitrogen level. So if blood ammonia level is increased and blood urea nitrogen level is decreased, then that implies it is Reis syndrome. Okay, if enzyme assay for UMPS, that is uridine monophosphate synthase enzyme, is decreased, that implies it is hereditary orotic aciduria. So that's about differential diagnosis. Now to summarize, we discussed about the introduction, that is autosomal recessive UMPS gene variation, uridine monophosphate synthase enzyme which is bifunctional, OPRTase activity and OMP decase activity. Fifth and sixth step, these are the steps related to orotic aciduria. Two types, type 1 deficiency of both enzymes, type 2 deficiency of only OMP decase. And features are accumulation of orotic acid in body and defect in DNA and RNA synthesis. Clinical manifestations, retarded growth, megaloblastic anemia, orotic acid crystals in urine, and urinary tract obstruction. Diagnosis, urine analysis for orotic acid presence, bone marrow screening for megaloblast, genetic gene testing for checking variations in UMPS gene, and enzyme assays for OPRTase and OMP decarboxylase enzymes. Treatment, cytidine and uridine supplementation. We discussed how this aids in the treatment. There are two ways. One, it revives DNA and RNA synthesis, and another, UTP, is an allosteric inhibitor of CPS2. Now other types of orotic acidurias, that is Reis syndrome and drug-induced orotic aciduria. So then finally we discussed about differential diagnosis. Yeah, so that's all about orotic aciduria. I hope that was clear. Thank you.